Hi, everyone. Welcome to Microbial Minutes, the weekly update on what's hot in the microbial sciences. I'm Julie Wolfe with the American Society for Microbiology, and today is the new Vaccine Strategies edition. There have been a number of reports in the last couple of weeks about different vaccine strategies, both on the antigen side and on the immune response side. And so I thought we would discuss these, but before we get started with the reports, let's take a minute to go over Vaccines and Immunology 101 on the next slide, slide 2. So here, if we're looking at the left-hand side, it reminds us that there are a number of different types of vaccine antigens that can be presented to the host immune system. That means it could be an attenuated version of a microorganism, or perhaps one that's been inactivated by heat or chemical treatment. Sometimes a vaccine might have only a component, only one recombinant protein that could be, for example, the outermost protein of a virus that would help to elicit an immune response should the entire microorganism then enter and try to infect the host. These different types of vaccines can be administered in different routes, and that the route of administration can also influence the type of immune response that is generated. So we're going to ignore the right-hand side for right now, and we're going to talk about the different types of immune responses that you might be able to elicit, for example, with a subcutaneous injection, like what we get with the influenza vaccine when we get a shot in our arm, or one that could be an oral vaccine, something like the polio vaccine that would have a higher effect against mucosal pathogens and in eliciting a mucosal immune response. And so we're going to talk about a new strategy for mucosal immune responses in our first paper, which is on slide 3. The title of this paper nicely summarizes the take-home message. This is a report from Nanoletters, a journal that is published by the American Chemical Society, and the title is Biomimetic Micromotor Enables Active Delivery of Antigens for Oral Vaccination. And so this is a new type of oral vaccination for eliciting a strong mucosal immune response. And what this is an ingestible microparticle that turns into a motor as it goes through the gastrointestinal tract. There are several different layers. The innermost layer is a magnesium microparticle. This is what also constitutes the motor, and that is going to be coated by titanium dioxide with a hole left on one side of this microparticle. As you can see in the orange titanium dioxide circle, there's a little bit that's been left uncoated, and that's important for the motor to be active. On the outside of that titanium dioxide, which is a relatively inert type of molecule, the microparticle is coated with, in this case, a red blood cell that is associated with a toxin. The toxin in this case is Staphylococcus aureus alpha toxin, which embeds into the red blood cell membrane and allows the presentation of this toxin in its natural format. So it doesn't have to be deactivated or mutated in any sense before it is presented to the host immune system during vaccination, or immunization, I guess I should say. The outside of this red blood cell membrane is coated with chitosan. Chitosan is going to help the microparticle to attach to the mucosa. It attaches with mucus and would help to embed the particle to the mucosal lining as it passes through the gastrointestinal tract. And then finally, there's an enteric coating covering the entire microparticle so that as the microparticle is going through the stomach, it is not activated and it is protected until that enteric coating is degraded as the particle enters the more alkaline part, the more alkaline environment of the intestine. And so what the authors are hoping will happen is that after the microparticle has passed through the stomach and the enteric coating has degraded away, the magnesium is going to interact with water, a natural part of many different constituents of many different areas of our body. And this is going to produce hydrogen bubbles that will propel the microparticle in a particular direction, hopefully in the direction of the mucosa, but also to help it to propel further down the intestine itself. And this should provide good exposure of that red blood cell membrane that also contains the toxin, and expose this toxin to the mucosal immune system to generate protective immune responses. This being a chemical or American Chemistry Society paper, there's a lot of material and verification that the microparticles themselves are generated in the way that they are predicted to be generated. So there's a lot of SEM micrographs so that you can see that the microparticles are indeed made in the correct sense, but then they did some proof of principle to show the effect of these microparticle micromotors within the host. And in this case, the host is a mouse. So mice were administered orally some of these microparticles, and we can look at the results in slide 4. Here they were given either the motor toxoid, so that's the magnesium covered in the titanium dioxide, but left with a little bit of a hole so that those hydrogen bubbles can be formed and the motor can actually be active, or those that were given a static microparticle. It's basically the same thing, except that these particles had no hole, so there was no part for the magnesium to interact with the water within the host. You can see, looking by fluorescence here, that the motor toxoid is able to traverse through the entire intestine at a much higher concentration than the static microparticle, which stays largely lodged at the upper intestine right after the stomach has released the contents into the intestine there. There are many different measures of immune response. In this case, the authors are looking at an antibody response, and of the different antibody classes, immunoglobulin A, or IgA, is most strongly associated with mucosal immunity. And so they looked at the levels of IgA against this Staphylococcus alpha toxin, and they saw that the mice that had been administered that motor toxoid, the one with the magnesium that was able to form those hydrogen bubbles, had much higher concentrations or titers of that antibody than those that had either not been administered a vaccine or who had been given that static microparticle. Incidentally, this is not the only microparticle administrative device that has been published recently. There was something that was quite similar, not in the motor context, but in that it delivers contents within the gastrointestinal tract, and that was in Science. That was more about delivery of insulin, so we concentrated on the one that was vaccine-oriented, but this is a trend that has been ongoing with delivery of many different biologics, and I think is something that will continue. Of course, you can find links to both of those studies down below our YouTube session. Go ahead and click those if you want more details. So we're going to return to Immunology 101 in our slide 5 here, so that we can talk about how it is that the immune system detects those different antigens, whether it be an inactivated or recombinant antigen, however it is delivered. Of course, the ability to respond to these different antigens is very complex. In terms of our B cells and T cells, these are very unique immune cells that have the ability to undergo different genome rearrangements, which is unique among all of our adult somatic cells. These B cells and T cells have receptors that incorporate various V, D, and J sections, and they can recombine or change around which of the different versions of the V, D, and J sections into either the B cell receptor, which will eventually lead to antibody production, or the T cell receptor, which would lead to T cell activation, in a way that allows our B cells to recognize 1 trillion different potential molecules, which is a huge number. But you have to imagine that these are going to have to recognize all of the different potential antigens that they would see, all the different microbial and allergic types of antigens, as well as those that would be from self, but that's another story. In addition to changing the gene order, there are other ways that the diversity is increased by changing single nucleotides within those selected VDJ rearrangements, and that's done through somatic hypermutation. So I have just some very basic illustrations of that shown on the right here. Now, antibodies are the basis for a lot of our immune response and our immune protection. Antibodies are vital, for example, in neutralizing pathogens, such as viruses, by binding to those pathogens, the outermost protein, and stopping them before they find their host cell receptor so that they can come into infect the cell and then reproduce and make more progeny virions. There are many different types of antibodies that have been characterized. In this case, we're going to talk about to start with, we're going to talk about HIV. HIV, there are many different naturally occurring types of antibodies, including those patients who produce broadly neutralizing antibodies. These are antibodies that can inhibit a wide variety of different HIV molecules, but they're very hard to elicit by vaccination. There haven't been any broadly neutralizing antibodies that have thus far been able to be produced by vaccine candidates for HIV. What this next group wanted to do was to take some of the VDJ recombination sequences that are known to produce these broadly neutralizing antibodies, put them into B cell lines now that genomic editing tools are becoming more commonly used. And so moving on to slide 6, we're going to talk about two different stories here, but both have to do with rearranging the B cell genes in order to produce different types of antibody responses. In this first paper, this is from eLife, the group took two different strategies using CRISPR-Cas9 technologies, and you can see where the double-stranded DNA cuts were made with those scissors. That's where they were cutting out different sections of the B cell repertoire, and then putting those into various cell lines. They were using the Ramos, it's a B cell line that's immortalized, in order to produce this broadly neutralizing antibody called PG9. And you can see in figure D here, that the strategy was able to produce some B cells that did have B cell receptors that were able to recognize this particular antigen. In this case, they're looking at the ONV protein of HIV. And what I'm not showing you here is that they were also able to see somatic hypermutation occurring within this engineered B cell line. So that allows greater affinity or potentially adaptation of that antibody within the host after that antibody sequence or the VDJ sequence has been selected and put into the B cell clone. Now, another group who has put their paper online on bioRxiv, but it is not yet has not yet been peer reviewed, so just keep that in mind, has taken this one step further. And we'll look at that in the next few slides. We'll go to slide 7. This group used a very similar strategy, taking different VDJ segments and putting them into a B cell line, but they went further and looked additionally at the ability to clone primary cell lines. So they took B cells from human patients or human volunteers and were able to engineer those B cells, which could then be purified by FACS, as you can see down, or I guess on the right-hand side here. Those B cells were able to make antibodies against RSV, influenza, EBV, and HIV, so a wide variety of viruses. And after the cell lines were grown for a few weeks, they collected antibodies from the supernatants and saw that the antibodies were being secreted into the supernatant by these different B cell clones as well. They then went yet one step further to show that these antibodies were protective. We'll look at that on the next slide. Here, just looking at A, B, and C from this figure, the group was able to add some of these cloned B cells that made RSV antibodies, and they put them into mice. They then looked at whether the mice were generating the antibodies, whether these B cells were active within the mice. And in B here, panel B, you can see that the mice that had been given the anti-RSV antibody-producing B cells did in fact have antibodies within their system, whereas the other control mice did not. And that these antibodies were protective because when mice were challenged with RSV, the RSV was not able to generate a wide variety of plaque-forming units, which were measured in panel C there, and that the antibodies were generally as protective as the antiviral compound, which was used as a control. So that's promised that these antibodies can be potentially generated from modified B cells put into a different organism than the one in which they were naturally produced or engineered, and then those antibodies produced by those engineered B cells can be protective. So this was written up. Both of these stories were written up in science-oriented outlets. Stat Magazine covered the CRISPR B cells in a very nice article recently, and the motorized pills were covered by the scientists. And I thought that the first sentence of this paragraph was particularly interesting. Traverso, who actually wrote the science article about the insulin, says that he hopes the insulin pill could be used to deliver all kinds of biologics and gene therapies, potentially well down the line, combining some of these different technologies, not only the motorized pill, but also the potential for using it for delivery of different genetic modifications for the host. And so that's going to bring us to our very last slide, where our summaries are that these micromotor vaccines facilitate murine mucosal immunity, and that engineered B cells protect mice from RSV. All of these studies have been done in mice, and that's important to note that these are extremely, extremely preclinical studies, and there needs to be quite a bit more work before any of this would be even close to getting into primates, let alone into humans. That's it for today's Microbial Minutes. Hope that you learned something. If you have any studies that you want covered next week, let me know by leaving a comment or a link down below. I'm Julie Wolf. I'd like to thank Ray Ortega for production, and I'll talk to you next time.